Linnaeus. And we really had kind of left out the scientific method as kind of a hanger. And we had left out the idea of evolution kind of as a hanger. And I did want to circle back around to those tonight, just so that we are okay with those. Maybe not necessarily to use the PowerPoint, but something we can talk about as a class. Did anything else from last week seem a little abstract, or did I need to go back and talk about any of the stuff from last time? I don't know, scientists, methodologies, really kind of more of an intro than anything too crazy, probably. All right. So this is a gen ed class, and obviously the idea of the scientific method is kind of important. And so maybe you guys can help me out. We can use the chat or whatever. You know, the scientific method's about logic, and it's about a stepwise approach to a problem. And I can throw this up if you guys want to see this. And it's not necessarily about using it for every problem that we've ever had, because obviously that doesn't really work. It's more kind of as a guide to say, all right, is this something logical and stepwise? Yes. Okay, then we probably need the scientific method. If no, so if this is, you know, what do I wear to school, or something taste and preferences wise, then in those cases, you know, the scientific method probably isn't really worth it or relevant, because a lot of that isn't about logic. It's about the things you want to do. You know, with clothes, it's like, well, can I wear it again, or is it really just too dirty to wear again? You know, does it make me look cute? It's not necessarily about how cold it is, how warm it is, what the wind is, you know, anything like that. So the scientific method really should be for the bigger stuff. You know, if you're trying to troubleshoot your computer, if you're trying to do lab work, if you're trying to work on the human genome. And basically, I think when we get at this, and again, if you guys want to text, you know, it all starts with a problem that we observe, whereby we see that it's complicated and complex, and where we desperately need a conclusion where we can attach what we really call a mechanism to it. So a mechanism is essentially the why. If you guys have nephews or nieces or children, and they constantly say, well, why? Rather than saying magic, because eventually we all have to go there, we're trying to give the why. And so when we talk about evolution, the why was really natural selection. The mechanism was natural selection. There were a lot of people before Darwin who talked about evolution. Lamarck is one of them. But since they were missing the why, they missed the mechanism, we never actually gave them credit. Now, it's not that they were bad or that they were wrong, but they were missing some of the scientific method components that we really needed. So you guys describe, or you guys see an issue, you're observing it. What's the thing you do next? What happens after we observe a problem and we're like itching to fix something?
Hypothesize.
Yeah, and so we tend to come back to the hypothesis portion. And I know folks really like to think of it as an educated guess. And yeah, you know what? There's nothing wrong with thinking of it that way. So, you know, with HIV, you can go back in the literature now after 40 years of seeing what happened. And you can see the educated guesses that the scientists made in the 70s and the early 80s, people like Gallo and Montagnier. And a lot of it was, gee, is it a virus? It might be a virus. Or gee, you know, maybe it's some kind of strange cancer. But eventually they got down to, I think it's this organism that is the cause of HIV. And so it went from more of an educated guess, which is still fine, to a tentative explanation. And a lot of the time it's stronger to have a tentative explanation than it is to have an educated guess, but it doesn't mean one of them is necessarily better than the other at all points in time. My thesis was about the Chesapeake Bay, and it was about organisms that were able to break down urea. So instead of saying, I think the phytoplankton dynamics are about the kinds of fixed nitrogen, we turned it around to say the availability of fixed nitrogen determines phytoplankton dynamics. And so once you have an overarching thesis or a hypothesis, the next piece that makes it scientific is that you can draw predictions and test them, right? So obviously, if I'm talking about fixed nitrogen, if I changed the kinds of fixed nitrogen, then I should be able to change the phytoplankton structure in the water, right? That's a prediction that I can make, and then I can validate that. I could be correct or I could be incorrect. Notice it's never true. And that's kind of an important piece. The null hypothesis is basically your experimental minus your control gives you exactly the same result. And so the null hypothesis is often the opposite of what you expect to happen. And I know alcohol is one of those common ones where folks say, well, drinking affects your reaction time, right? True, correct. But the null hypothesis would say drinking does not affect your reaction time. And so when you did an experiment where you had somebody drink and measured the reaction time versus someone who did not drink and you measured the reaction time, when you subtract the results from the control, you get a significant difference. It would not be zero. So sometimes the null hypothesis is like the anti-hypothesis. And generally, we don't talk about it much, but it is part of the steps here. And once this is formulated and once you're good with your hypothesis, the best part is testing. And I really think that's the part that we look forward to, although we probably need to be safe about it so we don't end up like Nobel, right? So we're looking for the mechanism. And I guess there's a couple things we should talk about, right? One of which is the idea of controlling a variable versus having a control. And I'm sure you've heard this a billion times. Whoever wants to jump in, what's the story with the control and what's the story with manipulating a variable at a time?
Well, you need to make sure that there aren't other factors that are affecting your results. So you have to set a control group to compare it to something.
So the control group is one that we don't manipulate. We don't amend, we don't change, we don't futz with. And the idea then is that in all of the other groups, everything has to be reproducible to the control. It's got to be the same dirt, if we're talking plants, same dirt, same light, same fertilizer, same water, except for the one thing that you're testing. So maybe your control is 25 plants and it's your stereotypical plot in your yard or your land. And then in one group somewhere over there, we're going to change the amount of sunlight, increase it by cutting down trees or decrease it by putting up shade. So we only play with the shade or the sunlight in that one. Everything else, the soil, the water, everything's the same. And to your point, if we mess with things other than the one variable, how do we know what actually caused the effect? Right? We really don't have any good way. The other thing is sometimes the measurements that you guys use or the outcomes that you're anticipating saying I was successful or not on can be very subtle. So in the lab, eventually you guys are going to have to do an unknown. It's called the unknown identification of the organism. And some of the changes biochemically, and these are going to be via color, are very, very subtle. And so without the control, you might miss a change, or in other words, a non-negative, because the colors were so similar. And obviously you're not running statistics on these. You're really comparing what you get to the control almost directly. So the control is really important. It's a basis to know if there's been substantive change. Now, on the idea of replicates, do you do the experiment once? I mean, when you think about the lab, you guys are doing it 16 times, basically the same experiment. And while the groups may get different numbers slightly based on how you chose to measure or what you chose to focus on or just how you perform the procedure, the results are comparable and the results are largely the same. So in that lab, we've got 16 different experiments, or if we're doing groups of four different replicates that basically support the overall hypothesis and conclusions here. In general, we like to see 3 replicates, but in the example of the plants that I gave you, plots of 25 are a good number because it really doesn't take that much to just grow some plants and water them, unless, of course, the thumb is less than green. I don't know if I told you guys the story, but I used to judge science fairs. And one of the most interesting ones that I had the opportunity to judge was my first one. And it was up in North Rockland where I was put in charge of, believe it or not, the 5th graders. And so I guess 5th graders are probably 10, give or take. And there was pretty much the usual stuff that you'd expect. A couple pyromaniacs, a couple volcanoes that were terrible. And I really shouldn't say that, but they weren't very good. And the winner was actually a 10-year-old girl who didn't have the best data, did not have the grandest experiment, but she was the one who followed the scientific method the most closely. And what I'm getting at was her replicates. So she was a hiker. I guess her parents would take her hiking during the Christmas break. My uncles used to do it too. So I get it. And like most kids hiking in the woods in the snow in the winter, your feet get cold, right? They're wet and cold and miserable. So she decided that the kind of sock that she wore would influence how warm her foot would stay. Okay, that's good. 10-year-old science, perfect place to start. So she rigged up an experiment. It was different kinds of socks she got from Walmart. It was a rock, some ice water, a thermometer, and a timer. And she did everything and made a graph and had a nice table. But what drew our attention to her was that she did it twice. Right? Everybody else, look, you can only burn something once. You can only light up a volcano once. She actually had 2 replicates, and despite the data being poor, she was the only one who did that. And so that was a big win for her, just from following the method to try and get the best results that she could. Other experiments, 3 might be enough. That was always our gold standard. For plants, it might be 10. You look at Mendel, he had thousands. And in general, the more data you have, in general, the stronger the conclusions could be if the data is good. Now, once you're done with your experimenting, you have to figure out how you're managing all of that data. And, we're obviously writing down numbers. We're obviously taking measurements. We're doing math or calculations or stats. But are you also taking pictures of your stuff? I mean, this is 2020 part 2. We have cameras. We can take video. You can actually show yourself doing the experiment. And those are really good things to do, especially with this, I don't know, mistrust in science today after some rather failed scientific publications got out there. One of the ones I'm referring to is the thimerosal one from 1999. Right? So he did a whole bunch of data mining, this physician. He wrote a paper. He wrote a conclusion section. The conclusion supported his hypothesis, and it said that mercury causes autism and that vaccines are dangerous. The piece that we all forget about is number 4 might be observations and data, but number 5 is conclusions and others doing your experiment, trying to reproduce your conclusion. And so the gentleman who, and I'm using that term loosely, he's really a criminal, who wrote the paper, published it, caused a significant stir in the community relative to vaccinations. And a few years later, was forced to retract the paper, admit that he had falsified the data. He was criminally charged and lost his medical license. And the reason for that is when people find a published paper and they cannot recreate the data, you start to ask a lot of questions. And the questions led them to an ethical inquiry that was not not won by the physician who wrote the paper. And so it's been retracted, but it is causing a lot of problems with vaccinations these days. And so if that person had pictures and videos and original sources and showed him actually doing the research, it certainly would have made it tighter. Although, look, a fraud is a fraud is a fraud. So just one of those things, when you're collecting the data, take pictures, create the evidence and the paper trail, write clearly, organize things into data, tables and graphs. And I'm going to recommend another thing. When you guys do the tables and the graphs, you always write a table legend, right? You always write in the results section, don't say my results are table 3. The results that are described in table 3 say this. And the reason why you go that extra step is so that you help people draw the same conclusion you did, right? The data still has to support it. But if you're in left field and they read it and they say, and you say what it means and they disagree with you, that's totally fine. But I would definitely lead the witness and I would help them understand how you got to the conclusions by describing your data to them, right? A graph or a table never shows anything, it describes, and then support the heck out of it with your discussion and your conclusions. And in the end, unfortunately, it's called research because this has to happen more than once. And if you think of it kind of like a spiral with X marking the spot in the middle, you're going to go around and around between your hypothesis and your experiments and your discussion and conclusions a lot of times, kind of like water going around the drain. And eventually, right, you hit that center spot. It might be 2 or 3 times. If you're Thomas Edison and it's the light bulb, it was 1,000 times, so the story goes. but eventually they come together and one will support the other. If not, then it's something you're either going to have to go back and tweak, or you're going to have to abandon until something else can shed some light on the challenges or the issues that you're having. If we go back in history, the gas laws were one of the first ones to go through this process. And I believe it was Boyle's Law, 1627, that started using the scientific method to draw theories and put them into play. So that's probably the shortest version of all time on the scientific method. It's familiar to us. We should probably be good with it, but I'm certainly going to stop and ask if folks have anything they need to ask or any questions about any of this stuff. I always ask a funny one, you know, are bar napkins okay here? Like literally like a back of a bank envelope. Is that cool when we do some of this work? I certainly wouldn't publish a bar napkin, but it would be wrong of me to say that I haven't had some amazing discussions at bars with random people who throw random ideas out that lead to epiphanies. So sometimes the numbers at a bar might not be appropriate to publish, but when you go back to the lab, it takes you down a road that gets you somewhere. So don't discount them. I certainly wouldn't publish them. I might tape it in my lab notebook and call it a day. But certainly those are valid experiences that might lead you to an idea, or I mean, maybe a theory or a law. So sometimes those outside the formal setting things are just as important or valid as inside the formal setting. And that's important to know, especially if you're going to go down the road of social science, humanities, you know, outside the world of medical science. Totally, totally, totally valid. Epidemiologists would probably do that often. All right, the only other thing in here is evolution. And in general terms, we're thinking about time and change. And as indicated earlier, the first tenets of this theory come up in 1809. Ironically, 50 years later is when Darwin writes The Origin of the Species. Ironically, 50 years after that is when we start having modern genetics. 50 years after that, we have the structure of DNA. 50 years after that, we have the human genome. Right? So it seems that the number 50, at least in human years, is a big deal. And in 2050, give or take, maybe it'll be 2030, that'd be nice. We're already seeing evidences of this with COVID vaccines, with hemophilia treatments. Once we have the human proteome, and we can make proteins and change proteins and get those back into the body, it's going to be a massive scientific breakthrough, the way the Human Genome Project and the way Morgan Genetics was. And so evolution really is about time and change. And if you read the tenets, they talk about the inheritance of acquired characteristics. So in other words, genes, they use the word traits. The word gene hadn't been coined yet. And the idea about, I don't want to call it use it or lose it, that was an earlier term. But the idea of pathways that are not used actually are not frequently transmitted from generation to generation. And so you really get more into the changes in frequencies of genes and traits from generation to generation based on what we call selective pressures. Now, in pure Darwin language, the selective pressure was actually, in fact, coined natural selection. So the idea is that there's a struggle. I would prefer to use the word competition, but struggle is probably the right word, where there's not enough space, not enough resources, not enough mates for everybody. And so if things continue on the way, some people, some groups of animals are going to have to go. And as a result of this selective pressure or this struggle, certain characteristics and traits are more favorable than others. Maybe before the pandemic, that was untrue, but during the pandemic, there are a group of people who are genetically resistant. That's a favorable trait. You'll probably see that trait passed on. Those who are susceptible will unfortunately have not been able to pass on genes, and you'll see those frequencies decrease in the population post-pandemic, 15, 20 years down the road. The classic examples of time and change and selective pressures really go into ecosystems. And we talk about the white moths versus the dark moths, and I have that slide here. Certainly the idea of the orange tiger versus the white tiger are good ones. The idea of giraffe necks are good ones. But let's talk evolution in microbiology terms, because why not, right? And last time I asked folks, how many of you guys have antibiotics in your cabinets? And why I don't want to see a show of hands, and I don't want anybody to out themselves. I would probably say a third of the students on this call tonight have some sort of antibiotic in their medicine cabinet, right? About a third, maybe half, maybe a quarter, but somewhere give or take. And I always ask the question why, because that means you did not follow the directions. And there's like that little federal thing on there that says you're in violation of the law if you don't use it as directed, blah, blah, blah. But more importantly, you're really applying a selective pressure that you don't follow through on. And so what I mean by that is you go to the doctor with a sore throat, right? Strep is a really good example of why we might show up. Oh, doc, it's terrible, right? And he or she chokes the daylights out of you with the tongue depressor. They get a really good sample. And most doctors that have done this enough times, by smell, know that you have strep, right? The physical appearance looks kind of like strep. Or they will do a rapid test and or an old school test on blood with a bacitracin disc. What they're looking for is Streptococcus pyogenes more times than not, or some other type of group A strep. And so right as rain, you get some amoxicillin or penicillin or whatever it is they're giving you, and you go home. It says take all as directed, finish medication completely. And so it's 2 or 3 times, we'll call it, for 10 days. Well, day 4 or 5, how do y'all feel? Yeah, better, exactly. And so as I'm seeing people confess, you feel better, you stop taking the medicine, or at least it's a lot easier to forget to do it. And when you realize 2 days later, it's like, I still feel all right, forget it. And in maybe half of those cases, you might get away with it, especially if you have a good immune system or you're otherwise healthy. But what happens when on day 7 or 8, you start to get that tickle back? Day 9 or 10, oh shit, I messed up. And then you have to go and try to take the medicine again, and you learn it doesn't work. So what you did was you had a population in there that was probably susceptible to the antibiotic you were given. There's probably a few strong ones in there, and there may even be a few antibiotic resistant ones in there. So you've got a mixed population with different traits that would lend themselves to having different favorability once you apply the pressure. That pressure is the form of the antibiotics. So here comes the struggle. Now they're being drugged. They're metabolizing the antibiotic into their cell walls, and they're becoming leaky and they're bursting. Your immune system sees this, it's hydrolyzing the weak ones, and the population decreases. But then you stop the attack. All the weak ones are dead. Maybe a couple strong ones survived. Definitely a few of the antibiotic ones, resistant ones survived. And now in days 6, 7, and 8, they start to multiply and become numerous again. They start to cause the same symptoms again. And they increased in abundance because those traits were favorable, the antibiotic resistant ones. And the ones that were weak, they decreased in number because you obliterated them. So you go back to the doctor and he or she looks and you play it off like, oh, I took them all. Oh, it didn't work. And they laugh because they know you're lying, and they pull out the biggest 750 milligram hard to swallow pill you've ever seen in your life. You ask for bubble gum, they laugh, you go home with horse pills. And the second time around, we generally tend not to forget because we pray there won't be a third time where we've got to do this. And so that's really a short-term directed case of really time and change, because we think of this usually on geological timescales. But biological timescales for us are very different than those microbes, which may have a generation time of 20 to 40 minutes. And so those days for them are like years and years for us. And that's why they're able to accumulate these changes over time. But here, the mechanism is natural selection. It predisposes that there are genetic differences, characteristics, traits in the population. And through an event, in this case, it was an antibiotic, it could easily be a climate change or something to that effect. The features, the traits, the genes, the characteristics we have will either make us more or less fit in the new schema. In the case of the bacteria in your mouth, the ones that were resistant were more fit. In the case of a rising temperature or rising sea level or more trees and pollen, we'll have to find out what that looks like. But disease-wise, it's very common for there to be genetically immune people. Think about the Black Death in Europe. A third died. The others were somewhat resistant. Think about HIV. People have mutations. They actually survive with the virus for years and years. So we'll probably see more of this. But the idea of time and change is really at the heart of Darwin's theory. And the mechanism that made it all work was actually natural selection. So with that, I'm done boring us with things we probably heard plenty of times, but I will do my due diligence and ask what sounded weird, what sounded different, what should we go back to, if any, and what kind of questions do we have so far before we look at culture techniques and microscopy. And I am recording. Looks like it's recording, right? It says it is. I hope, I think. So we're in good shape. We'll be able to play this back. Hopefully you guys can verify if it is or isn't. I think it is. So far, so good. It's interesting stuff. You know, we apply it. It's easy to kind of get at some of this odd stuff that happens. I live up in Orange. I had a water pump failure in my neighborhood, and we were getting water from the wells that was not treated. And it started right around March, so it was ironic it started around the pandemic. And when a pump fails, you tend to get an extra injection of chlorine and then no chlorine injected. And so the water in the house would smell overchlorinated, and then like there would be none. Overchlorinated, and then there'd be none. And I started with cramping and changes in output, shall we call it, back in March. And it would ebb and flow. So my body would try to clear it, and then it would come back, and it would try to clear it and come back. It finally took a month of antibiotics, a month. It wasn't even just 2 weeks of 2 different kinds of antibiotics to kind of displace the E. coli or salmonella that had moved in, and allow some of the ulcerations in my intestinal tract and duodenum and stomach to heal. Right? And where that 0157 come from, the one that makes us sick? Well, it was just a part of the natural community. And because of the traits that it brings with us, it became more numerous, and the stuff that's supposed to be in there became less numerous. And so, you know, we are microenvironments, and our bodies orify, and our skin and our armpits and the back of our knees and any folds we might have are these micro ecosystems where they have their own selective pressures, right? Deodorant technically is a selective pressure, right? You're putting the screws on the organisms in your underarms. So it's kind of interesting just how many different situations we can link this to in our daily lives. Now, I will also shamefully tell you that I had to provide more than a few samples for the doctors. That, you know, thankfully there's these little drop-off places at LabCorp where you don't have to talk to anybody. You just put them in their little bags and kind of, yeah, those types of things. And it took months to come to the conclusion. So once I dropped those off, one of the things that they were concerned with was that I had C. diff, right? And I'm sure folks that, I see some nods. Yeah. What do you guys know about C. diff?
You have to wash your hands with soap and water. You can't use hand sanitizer.
You know what, though? I think that's should be for everything. Hand sanitizer is not a good disinfectant, and it probably does more harm than good when it dehydrates your hands. So you're essentially opening a place for stuff to get in when we're trying to prevent it. But you're right, C. diff is very, very easily passed around. Hot soap, hot water, proper hand washing. And if you're visiting somebody, they normally give you a gown. Now, with all those nods, if you smelled C. diff, would you know it? Yes, we would. Right. So when the doctor said to me, you might have C. diff, I said, no, I don't. No. Well, how do you know? Trust me, it doesn't smell like that. Can we have a sample anyway? Ma'am, you can have any sample you want. You're not going to find C. diff there. And so what they do then is from the samples, they start to culture them, right? And so in order to be a classic biologist, you've got to identify the microorganism one of a few ways. We can do genetics analysis, takes too long, or at least only tells you what's there. It doesn't tell you what's there in any abundance. We can do a chemical fingerprint, or we can do it the old school way, like you guys were attempting to do in lab 2. And so we'll kind of slide over here to I think it's entitled, and I'm going to drop this in the chat for everybody, just in case you guys don't have this. I think it's entitled Tools of the Laboratory. Yep, there it is. And if you don't have it, let me just dump it in the chat for us. There it goes. And it starts off with cell culture. And you know, Beaker here doing whatever it is Beaker's doing. Strangely, it looks like a pipe bomb, I guess, is going to be our lab puppet of choice here. What we want to do is take the sample and culture it to find out what comes back. And so very much like your street plate technique, what we would probably do with some samples is dilute it first before putting it on a plate, spreading it out and allowing it to grow into colonies. And the colonies represent individual types of microorganisms inside that mixed sample. And so presumably stool samples have a variety of ecosystem type bacteria in there. Your intestines and your gut are in fact an ecosystem. And so we're trying to figure out if there's something that's more abundant than it should be, or if there's something that shouldn't be in there that's causing problems. And so what we typically try to do as classically trained microbiologists is grow it. And more importantly, we want to grow these things azenically. And so the idea here, azenically, is basically pure culture. And I think we mentioned that not everything can be grown azenically, right? There's things called complementation, you know, the old barter system. I have corn, you have wheat. You don't have to grow the corn, I don't have to grow the wheat, we can trade. So here there's microorganisms, and I think the classic example is I might make arginine, Alicia might make histidine. We both don't make it, so we live together and we trade it. This way you can do what you need to do with your proteins with my input, I can make my proteins with your input. If I separate us or members of a community that are built this way, you can't survive on your own, and often you don't get any growth. They're dead or dormant. So this doesn't work for everything. I have to be straightforward and honest there. There's some really cool advances where people use automotive washers and membranes so that we can stack cultures on top of each other, but the chemicals can still osmose and diffuse between the communities, just not the cells. So we've made advances in the last 20 years, but a combination of growing in chemistry or chemistry and molecular biology, so some DNA stuff, probably would be the choice of experts today, but it's always nice to know where we came from. And so here we've got the 5 I's, and probably you'll see this on the test. It's a pretty common question here. The idea of inoculation, incubation, isolation, inspection, and identification. And a lot of this is pretty straightforward, but there's some things that I do want to point out, and there's some slides that are a little more specific, but just kind of briefly, the idea here is that whatever the culture medium is, doesn't have organisms to begin with. And so whatever you put on there is essentially inoculated. It's an inoculum, you're inoculating. Just literally, by definition, introducing a sample into a container. Sometimes we're the container, but other times we're thinking strictly of Petri dishes and deeps and slants. I'd be lying though if I didn't say, you know, when you get done cooking, let's say chicken, it's probably pretty close to sterile, pretty close. The thing you inoculate it with first is probably the fork, right? And then the plate, because your fork and your plate obviously have more microorganisms on them than the meat that just came out of the heat or the fire. So the inoculation really could be a lot of different things. Maybe you make your own yogurt at home. If you went and got a Chobani or something with live culture, and you took a spoonful of that out and put it directly into a gallon of milk that you just opened, and you incubated it in a warm place, overnight, you would basically make a reproduction of the culture from the yogurt in the milk container, right? And that initial spoonful is your inoculum. Incubation really depends on the organisms, and we'll talk more about that. I think Lab 10 does the cardinal temperatures with you. So you talk about psychrophiles and mesophiles and thermophiles and extremophiles. For anything that infects the human body, 20 to 37 is the sweet spot. And generally, we're going to put this at 37 degrees because that mimics the human body. Not all organisms are going to grow at 37. Some have to grow less than 20. Some need to grow more than 60. Right? But obviously, this being a medically oriented approach to micro, anything that's in the mesophile range, 20 to 37 degrees, maybe even as high as 42 with E. coli, would be primarily what we focus on here. Likewise, physical factors could be salt concentrations, could be other pieces of the media that would be important. It might be CO2, right? Something might need to have a little bit of CO2 gas if they're a barophile. The salt is for halophiles, 20 to 37 degrees C. And so whichever you incubate at, make sure that it's the right temperature. True or false, the refrigerator in your home is an incubator? Yeah, there's a couple nods there. Yeah, you know, you have the best 4-degree incubator on Earth in your kitchens. 4 degrees is a temperature that some things grow, and in particular, it's psychrophiles. Most of the time, it's fungus, but certainly if you sit there with the door open, hoping that food is beamed into your fridge, and/or you open and close it frequently, the temperature is really not 4 degrees anymore. It's probably more like 10. And at 50 degrees, there are things that start to metabolize. They're not mesophiles, but they're things that certainly will spoil your food if you continue to eat them. My mother was a big fan of telling her children that things could stay on there for 2 weeks and we'd have to eat it or else. And when I moved away to school, I was doing the same 2-week rule, believe it or not. And I never was sick, and my mother definitely food poisoned me a few times. Her refrigerator in the office died and I ate it anyway. But it's just funny to see that a lot of things or a lot of places that we don't think of incubators really act as incubators. One of those being your kitchen sinks, your kitchen tables, your kitchen counters when you're getting ready to prepare food or a meal. Don't thaw your meat on the counter, right? That's the health department lesson there. Isolation is an interesting one. You have to have a way in your lab to take one colony or one species amongst the many that you're going to have to sift through. And there's a lot of ways to do this. Sometimes it's just using your eye and using a loop or a needle to be very surgical. Sometimes it's using specialized media to highlight differences in the chemistry of the cells so you know which ones you're interested in and you don't have to waste time guessing. Once you've done this, you're probably transferring it and growing it again to be sure that it's pure. You've got to do an inspection and an identification phase. And the inspection is usually visual. What does it look like? How many, what does it look like? What do the borders look like? How tall is it? Is it raised? Is it shiny? Is it smooth? Is it rough? Is it fuzzy? And I should be saying, is it rhizoid? Is it cuneiform? Yeah. Right? Use descriptive words. You guys understand what I mean. And then the identification piece is hard, right? You can't just look at it and say, aha. You might say it's a rod if you do the microscopy. No problem. What rod is it? You might be able to tell the gene by doing the DNA test and getting the result. Okay, no problem. You might even get the microorganism. But in likelihood, it's probably going to be a couple tests you have to do for identification. And it's one of those, after a while, you just kind of recognize it. Strep throat, people can smell. C. diff, you guys can smell. Some things have a look to them. And when you do enough litmus milks, or you do enough McConkey or EMB plates, E. coli will jump out at you like a strobe light on a police car. You'll know it's E. coli just from the flashing lights that it produces. Now, not actually flashing lights, but if I told you E. coli looked like fool's gold on EMB, right, that iron pyrite look, you know exactly what I mean, right? It looks like fool's gold, and only E. coli does that. It's a lactose fermenter, so you'll see heavy fermentation, cloudiness in the plates, and E. coli has a very particular smell. So if you start to smell it frequently, you get the idea, oh yeah, I know what that is, 100%. So kind of thinking a little more deeply about some of this. And this is part of the shame of doing this remotely, is you guys don't get a chance to actually put hand on plate on needle. What you're trying to do here is separate the colonies enough that you can get at individuals. So you'll inoculate it, you'll grow it, and then you've got to somehow spread this stuff out. Some of the methodologies that work are dilution. So you might dilute a water sample or a sewage sample or a stool sample, 1 to 1,000 or 1 to 1,000,000, just so that it's dilute enough where you get a fair amount of cells, but not too many. And if the plate idea is a little hard, think about the old mozzarella cheese in a cheese drawer, or think about the bread that you've got in the cabinet, right? Even before you start to see the speckles on your bread, if you took out a piece the day before, you took out a bun the day before, it smelled a little off and it might have tasted a little off. And even though you don't see the mounds of cells, you don't actually see the bread mold, you're tasting the hyphae. You're tasting the asexual reproduction pieces that are in there. The mold, the actual speckles that you see, are the sexual beings, the sporangia, the mounds of the new rhizopus cells that were there that only became recently numerous. And so while there's hyphae all over the bread, the colonies are probably 10 to the 9 of one particular cell or another. So when you dig an E. coli cell off of a plate, and the next picture I'll show you has this, probably 10 to the 9 E. coli cells on that colony. Whereas maybe throughout the bread, they're 10 to the 4 to 10 to the 6 cells, which is why you haven't seen them mound up yet. They haven't become enough numerous, or they have not had sex yet. And that's disturbing, I guess, considering you ate that bread the day before. But we're all here, right? And that also means don't think you can just throw that one piece of bread out and the rest is okay. You might eat it for dinner, but the next morning you'll know for sure it's definitely not okay. Now, the reason this works is we assume, and we shouldn't assume, we should trust but verify, that the colony has only one type of cell. And so when we pluck it off and we grow it and we plate it again, generally you want to make sure there's only one kind of cell there. That means the colonies look about the same, have the same about color, have the same kind of borders, have the same rasivity, and maybe even plus or minus are the same size. And when in doubt, or you're not really sure, then you can dilute it again and you can repeat the process until you only see one type of cell with the same color, the same shape, the same size, the same margins. In some cases, and I hate to throw wrenches, one of the ones that we use, and I'll throw it in here, we'll do italics maybe, nope. So pretend it's underlined or italicized. M-A-R-C-S, Marcessi, I believe that's the spelling, is called Serratia marcescens. This is an interesting critter. It was released in the New York City subway system in the 50s, aerosolized, to see how a biological agent would travel through the subway. And this particular microorganism, when it grows, has a very fishy urine aroma. So when you stop at some of the subway stops and you smell it, it's not just the folks who live there, it actually could be leftovers from the 50s. I mention this because up to 28 degrees Celsius, Serratia will grow a pink color. At 28 degrees, there's a shift in metabolism. So one gene turns off and another gene turns on, and the colonies become white when they're grown at 28.5 or greater. And so sometimes you'll look at a Serratia plate, and maybe you'll even see one in the lab, and you'll see pink and white colonies, or you'll see pink colonies with slightly pinker centers, or you'll see beige colonies with pink centers. In that case, it could just be a difference of expression, and that's why we might actually take a cell and transfer it more than once to be sure that it's pure. And what that basically looks like is this. Right, here's a plate where we have a mixed culture, and we've got something mixed up with bacilli and cocci. So it might just be E. coli and Staph aureus, right? Simple, straightforward. We mixed it up, and we're giving it to you to separate. What you're seeing underneath these scratches in the surface of the agar are really streaks. And so you took a little bit of sample, and you started heavy here, and by the time you've zigged and zagged across the plate, and you're starting to get more open and more and less frequent with your strokes, you've diluted the cells enough where you start to see individual colonies. And so these colonies grew white, these colonies grew yellow. And so I would say the two on the left are definitely the same organism. The three on the right are probably the same organism. And with a needle, we would touch ever so gently into the colony, and transfer it to a new broth tube, or touch gently here and transfer it to a new plate. Touch this one, transfer it to a separate plate, and make sure that on the one plate you just have the yellow colonies, on the other plate you just have the white colonies. I'm not going to lie, fungus will grow sometimes. When you see fungus, some people call them Fred or whatever, they're very obviously not either of those two kinds of colonies, and they'll usually be white with black fuzzy edges or green with black fuzzy edges. The fuzzy, the rhizoid nature of it tells you it's a fungus, and it's not what you were looking for. Not all fungi look like that, but oftentimes the contaminants are fungi. All right, so isolation. Media is kind of interesting. And so here's where some of the chemistry comes in that makes it real handy. The media is really the nutrients and the support for the organism's growth. When we use agar, the agar or the matrix that the agar creates isn't actually a source of nutrition. It's solely there to put a framework for the organisms to live in. So agar isn't digested. Agar is just a piece of the media to provide solidity and some sort of structure. Generally, though, every growth medium is going to need water and some sort of carbon, nitrogen, hydrogen, oxygen, phosphorus, or silica. So CHON, the big four, phosphorus and or silica, depending upon what you're trying to grow. And when I say some form of carbon or some form of nitrogen, what I'm getting at is what kind of oxidation state is the carbon, the nitrogen, the oxygen, the phosphorus, the silica in. Nitrogen could be nitrate, it could be nitrite, it could be urea. And so some organisms like nitrate over ammonium. Some things don't like ammonium at all, and it's actually toxic to them. So when you make your media, you have to be cognizant of what am I trying to grow versus what do I have on hand. Vitamins are needed for some microorganisms. They don't all make their own. B12, cyanobalamin is one of those big ones, right? And if you guys take B12, it comes from a cyanobacterium. So in nature, I could just add cyanos, right? Makes a lot of sense. Or if I'm trying to do this in pure culture, I actually have to add B12 or B1, niacin and thiamine. So B6 and B1 tend to be other ones I have to add. If you're doing this, you add the vitamins and antibiotics and anything like that after you autoclave it, after you sterilize it, because things that are heat sensitive do not live in the autoclave. Trace metals are another funky thing, right? We need iron, we need titanium, we need chromium. So do the microorganisms. And if they take iron that's in +3 form and you give them +2 form, that might actually be a problem. So the form actually matters for each microorganism, but as a generality, those are the things you have to have. You might need to supplement some CO2. You might need to take all of the oxygen out of there entirely if you're trying to grow an anaerobe. pH is definitely important. Most of these things are going to be pH'd to 6.8 to 7.2 or 3 or 4. And if the organism produces a lot of acid or a lot of base, you may have to add a buffer. Now, just from a nerdy scientific point of view, anybody remember what a buffer is about?
Bringing like homeostasis back, or trying to.
Helps increase the reaction.
All right, so we've got some good ideas out here. Buffers, I think, are a little more closely related to homeostasis in that we are trying to resist a change in pH. So things that are buffered, think about your blood, for example. Everybody coming out of A&P should be able to talk about the carbonate buffering system, right? So we've got carbonic acid that turns into bicarbonate. And if we push it further, it turns into carbonate. And so essentially you're adding conjugate acids and conjugate bases in a buffer so that when an organism makes acid, the conjugate base will neutralize it. When an organism makes base, the conjugate acid will neutralize it. And this way we can maintain the pH for as long as we can. Eventually the buffering capacity, especially in lab media, is used up. And what you'll see is the cells may clear, the culture may clear, which is indicative of cell death. It may enter a dormant state because the conditions for growth are not favorable, and in which case all you have to do is do a transfer. But these are things that typically we have to consider. Obviously, the incubation piece, temperature comes into that as well. Generally, we have a lot of choices when it comes to media. You know, if I'm just growing something in general purpose, I probably just use a nutrient broth. Sometimes in the lab, I would grab something called LB, and LB stands for urea broth. Largely, it's just peptone and beef extract. Smells like M&Ms until you mix it up, and then it doesn't smell very good at all. But whether it's nutrient broth, nutrient agar, something more more enriched, it's all going to have C, H, O, and N with the vitamins and the other cool stuff that we need, depending upon how fastidious something is. Basically, how strict are the nutrient requirements? Or in general terms, how much of a pain in the ass is it to grow? The more fastidious an organism is, the more enriched, the extras that you have to add to make the media supportive of the nutritional needs. And so, as we said, you know, agar is a solidification event. It's a polysaccharide we get from red algae, and the organism never digests it. It never eats it up. It's simply added to the medium while we're boiling it to make it so that there's some structure. The organisms can grow on top or it can grow within, depending what we're trying to do with it. And the autoclave, it stays warm. It doesn't start to solidify until 45, 42, 40 degrees. And at which point, once it's solidified, it sets up like literal jello. If anybody cares, the math works out to about 1.5% agar in anything that we make, including the plates that we make in the lab or the blood plates that we buy. And where we need to, we can heat these back up to about 55 Celsius for some experiments that are kind of cool to liquefy it. Just be ready to go because it will solidify again once we get down to 42. So again, the agar is put in for structure, but the agar is actually not digested. And what you'll tend to see on old plates is the agar remains, it just dehydrates. It doesn't actually disappear. So in terms of media, we can kind of group these then into 4 general types. Synthetic, in which we know exactly everything that's in there, all of the organic, all of the inorganic stuff, not only the formulas, but the exact ratios. So if it's something I'm making, you're going to see 5 grams of this, 2 grams of this, half a gram of that. A complex medium might not say half a gram of this specific compound or half a gram of this specific compound, but in the earlier example, it may say 5 grams of dehydrated beef broth or chicken broth. What exactly is that? Well, we have an idea of what it is, but I couldn't tell you the exact chemical makeup with the exact ratios of the compounds. It's certainly what we need to stimulate and support life, but there's some stuff in there that's not definable. Kind of like whiskey. If you guys like whiskey or derivatives, 210 compounds, I can tell you what 9 of them are. The rest of them are just like things that we can characterize, but we don't know exactly what they are or what they do. All right, so synthetic is something that's synthetic, synthesized with known ingredients. Complex, a lot of it is hodgepodges of organic stuff. And if you can get away with complex or non-synthetic, the costs are much lower. Sometimes you do, though, for fastidious things or specialized things, you need to go up to the synthetic. General purpose stuff grows a broad range of microbes. LB for everything. E. coli would grow in the bumper of your truck. You don't need anything magical for E. coli. Even glucose is more than enough. But if you want that strep to grow, to find out if that's what's in your throat and that's what you have, then you're going to need to enrich that medium with blood. And so blood plates are a very classic example of enriched medium. 5% defibrinated sheep's blood goes a long way in supporting metabolism. Sometimes we use yeast extract. So rather than something defined, it's just yeasts that are boiled, digested, and then turned into a component of the media, which probably has a bunch of fungal things in it, a bunch of fungal metabolites in it, but it does the job, and it's enough for things that are somewhat fastidious, like alkaligenes faecalis. Let's see. Show you a couple pictures here. Has anybody ever seen this? I know there's no label on here, shame on me, but have you guys seen this yet or have an idea of what we might be seeing here?
I don't think so.
No.
So these happen to be blood plates. And these are blood plates that don't look quite so red. They've been inoculated and incubated. And on the top, what you're seeing is the streps that are doing hemolysis. So the alpha hemolysis is the green, kind of this last streak here. It's a partial digestion of red blood cells. These up here where you could almost read through the medium, that's good evidence of beta hemolysis. So a clear or a full lysis of the red cells. And then really, and I don't want to call that on the bottom, what we've got is gamma hemolysis, which will tend to look gray or white. And maybe at the edge, it looks that way. Maybe. I don't want to make claims about these here. But gamma hemolysis is the absence of hemolysis. And so that greenish kind of tinge here, and the color is a little off in fairness, would somewhat resemble with alpha hemolysis bile, if you've ever seen the color of bile salts. Because bile, much like alpha hemolysis, is a partial degradation of red cells. So if you've ever seen a strep test, it will look similar to this. The strep pyogenes, I should remember what kind of hemolysis it is, would have a clear zone around the antibiotic disc because they are susceptible to bacitracin, and you would see hemolysis on the rest of the plate. Now this is good for yeasts, some fungi. This is great for streps. But there's a whole host of other specialized mediums that we might use. And this is where, or this is one of the areas where it gets a little complicated. Selective media will typically allow one group of microorganisms to grow while inhibiting or partially inhibiting a different class. So one of the ones that we tend to use in the lab is crystal violet plates. And crystal violet is a stain that's added, and it partially inhibits one type of microorganism while allowing the other type to grow. Do we use phenylalanine? We might even use phenylalanine plates. And so what you'll see, I'm just going to make one up. Let's say we have a compound, we'll call it compound X. We add it to a medium, and it inhibits gram-positive cells. So what you'll probably see is E. coli's and Salmonella typhimuriums and Pseudomonas aeruginosus. Gram-negative things will grow in abundance. But the Staph aureus that we'd inoculate with it as a control would show scant, if any, growth, right? Because that compound X inhibits or partially inhibits the staph, but it does nothing metabolically to the E. coli or the Salmonella. And so selective medium is typically when you have a mixed community, but you're trying to screen it for something in particular, like a gram-positive or a gram-negative. Differential medium is media that will allow things to grow, but will show a color or a media difference based on the differences in biochemistry. So one of the plates that we use is something called mannitol salts agar. Mannitol is a sugar. It's a funky sugar. Not everything can ferment it. The salts are about 7.5%, which means you need to be a halophile, something that likes salt to grow in 7.5% or greater. And so let's pretend we're going to put E. coli, and we're going to put Staph aureus, and we're going to put Staph epidermidis on there. So 2 staphs and an E. coli. The mannitol salts, in particular the salts, don't get along with E. coli. High salt, think about beef jerky, think about preserved meats. High salt destroys E. coli. And so on our mannitol plate, you're not going to see E. coli growth. The 2 staphs, though, will grow because they are halophiles. Now, the mannitol part of this is the differential piece. Staph aureus will turn yellow, and the plate will turn yellow in a mannitol plate if Staph aureus is present. Staph epi will still grow, but it stays red because it does not ferment mannitol. So we can use the plate differentially if we want to figure out which staph is Staph aureus. We can use the plate selectively if we want to differentiate between E. coli and staph. Or we can use it both selectively and differentially if we know we have a mixed community that includes halophiles like staphs. Mannitol is a good example of that. McConkey is a really good example. It has lactose, it has an indicator in it, and it allows you to select for gram negatives. So a gram positive like staph wouldn't grow. And it allows you to differentiate between lactose fermenters. So in other words, in the way we use it in the lab, that differential medium, the McConkey, would allow you to determine if you had E. coli, Salmonella, or Enterobacter aerogenes, just how it fermented or did not ferment lactose. So these are very helpful plates and very helpful kinds of media for microbiologists who are doing kind of specialized things. If I show you a picture of it, notice here's a mixed sample. On the left here, this is grown, I'll blow this up for us. This is grown on just general purpose medium. And notice there's heavy growth here. If we take the same mixed culture and we put it on selective medium, we're clearly partially or fully inhibiting some organisms, as not all the colonies on the left will be able to grow on the plate on the right. And in a differential setting, right, all the media grows, or all the organisms grow like we saw above, but some of the colonies in this case are colored differently based on the biochemistry and the metabolites they produce and how they interact with the chemicals in the plate. Right. Sometimes it's the medium that changes color. Sometimes it's the colonies that actually change color. And so what we're seeing here on the top is actually a mannitol salt plate. And where you see the red, so this is probably like Staph epi. This is definitely not Staph aureus. This is indicative of Staph aureus. This is indicative of Staph aureus. That yellow coloration and the yellow discoloration of the media is really what you want to positively ID staph on mannitol. This red over here, even though it's growing, you know it's a halophile, but it's not Staph aureus because it doesn't ferment the mannitol. It doesn't turn this side yellow. Right, down here, what we see are probably at least two different kinds of colonies. You've got the red/beige one. You've got the yellow/beige one. I don't know. I'm not qualified to tell color. And so certainly there are at least two different kinds of cells here. I'm probably going to guess three, because I would say this is one, this one is two, and these very large colonies with strange borders would be number three. Even though the colors are similar, don't just go on the colors. Look at the border. This is round. This is smooth. This is pretty uniform. These are not. So there's probably three different kinds of cells here. And with your loop, you would be able to pick each one of them off, knowing you got something different, and inoculate them into new tubes. Let's see. So there's a couple other kinds of medium. I will show this to you just because this is one that you're going to see. These are carbohydrate fermentation tubes. And what they typically are some sort of sugar. We use lactose, dextrose, and sucrose. And we add a phenol red indicator to it. Inside those tubes is a little baby test tube turned upside down, and it's called a Durham tube. The point of the Durham tubes is to collect gas if there's any fermentation. And so the left would be our control or a negative. It's a red tube with no gas of any kind. And when you guys look at these in the lab, any red gas carbohydrate tube is a negative. Don't even look for gas. It's just negative if it's red. The question will probably be, okay, what is it negative for? And it's negative for fermentation. In the middle, we see yellow. And the reason why I can tell you the middle is an acid tube, it's not positive, it's acid, is because that organism had the ability to ferment the sugar. And we know that fermented products are typically acid products. This one also shows yellow plus a bubble. So when you see yellow, definitely look at the tube and find out if there's gas in it. And this particular tube has gas, so we would say this is an acid-gas reaction. Sometimes, like Staph aureus, only gives you acid tubes. And so when you do your unknowns, you're going to look at sucrose and dextrose and lactose, and that's where I would start making my identification of the unknown. 3 acids is almost always staph. 3 negatives, you got about 2 culprits. And so by using the sugars, you can really reduce the whodunit quickly at the beginning, right? And so I'm giving you the hints because it really doesn't need to be an anxiety-driven process. It's just one where you got to take your time, take a look, make sure you see what you see, make some good notes, and then compare it to a list of known values. We don't stick you out there with no guidance. There will be some known values there for you guys. And so with that, good place to pause, good place to ask questions. You're going to see these plates again in the lab, so it's not like it's one and done. So that's a good thing. You've already started to do experiment 2, as I understand it. And so the separation of mixed cultures should look somewhat familiar. Probably after you do that, you're going to be doing some microscopy. So you'll probably see some staining. You'll probably have discussions about gram-positive and gram-negative. So you'll look for things that are purple, or you'll look for things that are pink. Maybe you'll talk a little bit about acid-fast staining before you get into the selective and differential stuff. So we've got enough time and enough distance to do it, to think about something else, and then to come back to it. What are you guys thinking? That was a good chunk of stuff in a couple minutes there. What kind of questions do we have? What do folks want to ask? Practical questions, I can tell you that selective and differential comes up. And a lot of the times you'll see a plate and you'll be asked, is it used selectively, differentially? And it never says or both, but or both could be an answer. So on that staph plate, you wouldn't say differential or selective, you would say both. Right? If only one side grew and the other side didn't, you would say selectively. If both sides grew, but one was yellow and one was red, that would be purely differentially. And so sometimes, and this is the challenge moving out of A&P, you know exactly what you're looking at, but you have to answer a question that's hard to understand. And so that review sheet at the back of the syllabus really talks about selective and differential, and it identifies a variety of plates for folks that I know for a fact that you'll probably have a good chance of being asked. And for that reason, they're definitive tests for an organism, which is why I've mentioned it.
When you said about answering that the question or the answer can be both, but it won't ask that in the question, but we would know that we can put that as an answer. That's what you're saying.
That's what I'm saying, yeah.
Okay, thank you.
Yeah, sometimes you get a plate and it'll have 2 or 3 things on it. And you've really got to sit there and ask yourself, what am I looking at? Now, if we were live, you'd have the plate, you'd be able to pick it up, you'd be able to smell it, unfortunately. You would see what's written on it. Doing this in the remote format, you'll still see the plate, you'll still see what's written, how it's labeled, what's on it. You just don't get to look at it in your hand. You don't get to smell it. So you're going to miss some of those touchy-feely cues that you would have the advantage of in the lab, just not the advantage of over Zoom. And that is kind of a bummer too, because that is part of the experience. You haven't lived until you've spilled E. coli on yourself and you've got to clean it up. Oh, I only saw a couple of folks look disturbed. That's not healthy there. Come on. I had one semester where, and this is again hard to kind of illustrate over Zoom, especially with, you guys can barely see that. So in the lab, normally you take things off with your pinky, right? You don't use this finger, you don't use this finger, because if there's anything contaminating the cap, you'd much rather lose your pinky than your thumb, right? And so we always pick it up in the pinky and it always stays in our hand. We never put this back on the table. We never lay it down. It's a big no-no. So you start to learn to juggle tubes with lots of stuff in them. Unfortunately, now I've got a test tube with no cap. And the next thing that you guys would do would be to pass it in front of the flame. Well, there was a student who got too close to the flame and instead of keeping it pointed away from herself, pulled her hand away from the flame, pulled it back in that motion, dumping E. coli down her blouse. And it happened to also be test night. So the student knew full well that she didn't want to miss the test, but now we had a major spill situation. And quite frankly, a shower was required in this instance. And so that's some of the lab that you guys aren't catching either. And I'm sure you're probably relieved that you don't have that opportunity to pour E. coli on yourselves, but it does kind of make it real, right? It does kind of, it's part of the experience. Like when you guys cook and you burn yourself, it's not fun, but it's part of the experience. And so I hope nobody spills E. coli on themselves. That wasn't the lesson there. But you know, sometimes it's a little strange doing it this way. And so I'm sure all the hints are appreciated. And if they're not, tell me and I'll stop. All right. Does anybody need to walk away for a couple minutes? The microscopy piece isn't really long. I assume folks with the screens off are kind of bouncing in and out. I think folks are doing it anyway. And I am recording this, so should we just look at microscopy, or what do you guys want to do? We have an hour and a half, and I don't think we need it. So otherwise, we'll end early, and that's probably a good thing too. Let's do it. I like it. You don't have to ask me twice, guys. Like I said, trying to make this a little easier. I know this isn't ideal, so anything I can do to help with that, I'm trying to do it. All right, so we go to microscopy, and it's as fascinating of a field as anything else. And I'm going to show you this quick, and then I'm going to come back to you guys, because I want to talk about microscopes in the respect that microscopes are tools, right? They are something that you use to help you get a job done, like a knife, like a screwdriver, like a hammer. I see in the lab too many students get entirely too frustrated with the tool, and it really becomes like something eating you guys away. It's just a tool. That's all it is. You guys are better than that. And they all basically work the same way. So if you had an A&P Live or you had a general bio, even better, you probably got familiar with looking at a lot of slides. Part of the micro experience was making your own slides. And hopefully we get to do that with you guys one day. Part of the experience is putting a slide upside down on the microscope and saying, shit, I can't get this to focus. Or sometimes part of the experience is putting a slide on there and asking me what the Turtox organism looks like, right? And if you had a live class, the dreaded Turtox is actually the name of the manufacturer. It is not a real organism. So if you guys fell for that, I'm sorry. But again, part of the experience. And so learning how to use a tool sometimes is as important as what the tool can do, right? Knives should be sharp. They shouldn't be dull. That's how accidents happen. The microscope should be clean. It should be organized. It should be put away a particular way. The lenses should be wiped down with lens paper. We don't want to leave oil on it because that messes it up for the next person, right? So all of the things you would do with your computer, your phone, well, maybe not your phone. That's probably a bad example. But tools that you take care of, wrenches that you degrease or what have you, knives you sharpen, that's what you do to a scope. And I'm sure we've used them enough to know that we're not only making things bigger, right? We're not only magnifying stuff, but we're also trying to increase the resolution, right? We're trying to zoom in and not have something be pixelated and blurry. We're trying to zoom in and actually see some detail. And so if we use your cell phone cameras as a good example of this, right? You have optical zooms, you have actual zooms, and then there is some processing that happens in the background. And the processing in the background usually sharpens edges or brings detail more clearly into the picture. And so if you think about how the camera on your phone works, part of it is the lens, it's the concaveness of it, it's the curve that allows the magnification to happen. Sometimes it's the quality of the glass or the sapphire that we're using. Certainly the higher quality, the more polished it is, the better it's going to work for you. And what you're trying to look at also will dictate what kind of magnification you really need, what kind of microscopy you're going to have to do. In A&P or Gen Bio, we stopped you at the 43 objective. The 40 objective with the eyepiece multiplies 400 times. And for A&P, that was more than enough to see your cheek cells or an epithelial cell or an egg under the microscope. Well, that's fine, right? Because some of those cells were like 500 micrometers, they were 0.5 millimeter, they were 1 millimeter in size. E. coli is about 2 microns. And so we need to do a little better here in terms of getting you magnification and getting you the resolution you need. And so in the micro lab or in a lot of the other labs, there's a 4th objective. It says 100x on it, and it also says oil on it. And we use an oil immersion lens to get you the extra magnification, but also bend light so that it doesn't escape to the sides, bend it so that it curves back into the objective and it reaches the processor, it reaches the sensor, your eyeball. And so this is pretty much what the lab microscopes look like in microbiology now. And we've got your eyepieces. These probably magnify at 10x in the lab, very much like the standard ones do. It has an arm for carrying. It's got the body where we attach everything to. The nose piece with the objective lenses, typically scanning at 4, low at 10, high at 40, and oil at 100. So 100 at below plus 10 up here gives us, or I should say times the 10 up here, gives us 1000x magnification, right? And on this microscope, which is just your light microscope, or sometimes you might call it a bright field scope, this is pretty good for most of our purposes. One of the big tricks is figuring out how to do the focusing. These microscopes are typically called parfocal microscopes. And so essentially what that tells us is if you get something to focus in low power, it will be in the same focus at high power, and more than likely with fine adjustment in focus at oil resolution, parfocal. The other interesting thing here is, we have the tendency to put the light all the way up here with the intensity and then open the diaphragm here wide open. And look, the microbes are small and they're really transparent even with stain. Sometimes the biggest trick I can tell folks is reduce the amount of light. Lowering the amount of light opens a whole world of microorganisms that you would not have seen if you left it all the way jacked up. And I know it's kind of hard to show this kind of stuff to you because we are remote, but trust me, just file that in the back of your mind. Reducing the light sometimes opens up a wide of microbes that you never wanted to see before. A couple other just quick pieces, because I know we all know how to use this. Coarse adjustment is only on scanning power. If you use the coarse anywhere else, it may not be catastrophic, but it is a way to smash the objective into a slide. And so obviously we don't want to scratch the optics. And so once you go above scanning, above that 4x objective, we only use the fine adjustment knob. And so just food for thought there. Now, typically, and there is some physics behind this, and I obviously don't want to spend a lot of time on the physics because this is not a physics class. The light source comes out with however much light, and light moves obviously as both photons and as a wave. It's going to hit the specimen, and a lot of the times the beams are crisscrossing. So they're going to kind of converge and then diverge as they go through the condenser lens. They're going to hit the glass specimen. They're going to start to pop out again or diverge. The objective lens focuses it again, and we kind of go back and forth at it diverging and focusing between the different lenses and the different mirrors that are in the microscope. And what tends to happen is the picture that you see is usually a mirror image of what's actually on the slide. So if you're looking at something moving and it looks like it's going left on the microscope, you're going to need to move the stage to the right to keep it in focus. So that's one of these things that we sometimes tend to forget. Occasionally, you might have done the letter E, where you look at it and it looks right side up, but underneath the microscope, it's upside down and backwards. Right, so it's just a reminder that there are some mirrors in here, and the image that you're seeing is not the actual image that's there. It's a projection with a little bit of processing. So now think about resolution. Notice all of these beams of light are coming out. They're being captured by the objective and the specimen. They're being captured by the ocular lens and obviously making it to your eye for integration in the brain. What if some of these weren't being caught? What if some of these beams were going out to the sides and they were missing this objective lens? Because more than likely, we are having some of that happen. Well, the answer there is you start to lose resolution, or the things that you're looking at look less detailed than you think they should be, or really than they ought to be. And so here's an example of resolution in conjunction with magnification. So what you're looking at is a louse, might as well be a flea. And on the left, we've got a scanning power versus something on the right with higher magnification. And you can see the level of detail, the amount of resolution in difference between a lower power objective and a higher power objective. The left is really good to find stuff, right? And that's the idea of why we scan, just so that you get a good field of view. By the time you go up, you find something, the field of view gets so narrow, but the magnification and the resolution increase. And that's why we tell you, hey, let's start on scanning. When you find what you're looking for, that's when you go up in power. And again, being parfocal, once you have it focused in scanning, it should be pretty darn close in low, high, and oil, right? You'll only have to make those super fine adjustments a quarter, a little click left or a little click right. Now, for those who wonder why oil is so magical, oil is about resolution. And so on the left, you see the slide, you see the beams of light, and you notice we're losing a couple of those pathways. So when we lose data, you lose resolution, right? You're losing information that would otherwise be helpful to you. It's not getting to the eye. And so on the right, we add oil. Now, the first thing you notice is, wow, that's a lot of oil. The second thing you notice is, wait a minute, the lens is in the oil. Yeah, it's called the oil immersion lens exactly for that reason. And so the oil acts as an agent that changes the refractive index between the glass and the air. And so you actually bend more light toward the eye than would otherwise scatter without the oil. And so you not only get greater magnification, right? It's like 600 times more, but you get much greater detail because more of the light makes it to the detector, right? To your eyeball. So the oil lens is very helpful, especially when you're looking at microorganisms. A flea, an egg, tissue cells, nah, I wouldn't bother. But when you suspect a rhizopus or a streptococcus, yeah, this is a really useful tool, especially if you can tell on high that it's cocci, but you're not sure how they're arranged. Going up to oil will probably solve that for you. And you'll get to see a lot of the detail, whether it's chained or clusters or tetrads or sarcinas, clusters of 8 or more. You'll be able to see that very well under oil versus other magnifications. And in fact, when you look, right, this is kind of a scanning perspective. This is not only magnified, but with some extra detail and resolution. And even in these bacilli, you'll notice that you can see these little oil droplets or these little inclusion granules that there's no way you would have seen in the other shapes or forms. Right? It's really nice to be able to get in there and see, oh yeah, look at that, it's spirilla, sure. Here's some bacilli, no problem. Here's some cocci, awesome. Here's some vibrio, there's some bent rods here. I wonder what this might be. And sometimes microscopic investigation might be enough, right? If I saw chains of cocci and I smelled strep throat, I would give you antibiotics. If you had diarrhea and I looked at the water sample and I saw those curved rods, I'd probably be asking you where you were drinking from because you might have cholera. Are you guys campers? No. I went out to Montana years ago, had a friend, he was getting married, didn't really tell us that there was a wedding until we got there, which was hysterical, actually. Then I saw his wife and it was even more hysterical for other reasons. But the honeymoon was at this place called East Rosebud Lake. And if you know it, it's beautiful. If you don't know it, it's a nice place to visit. The water in the house was not turned on all winter. We turned it on and it was gross. It was rusty. There was a beautiful mountain lake just down the hill from the cabin. And I went down there and I grabbed a bunch of water. We were making pasta and ziti in the house, but I needed to make sure that I boiled it first, right? And no matter how clear and how pretty it looked, there was definitely giardia and other types of things in that water that would have been no good for us. So naked eye, no good. Scanning, probably wouldn't have been able to tell. But zooming in, you would have absolutely seen the giardia or the amoeba or some helminths, some nematodes in there if you had a microscope prior to drinking and or boiling it. The beautiful thing is once you boil it, it's just extra protein and carbs for you to ingest. So it's actually okay and not infectious afterwards. Kind of like if
So
you,
I have a,
go ahead.
I'm sorry, not to cut you off. I have a stupid question. So like, say if I go to like a lake or something such as that, and I'm in the water, can any of these things, like, can I get sick from any of these things?
So the answer is maybe. Are you drinking in that water or is it just splashing on you and you're kind of spitting it out and wiping it off?
I guess I would say spitting it out and probably wiping it off.
So the maybe is because many of these organisms have a threshold for inoculation. So for example, if you don't end up ingesting a total of 10 to the 8 E. coli, you only ingest 50,000. You're probably not going to get sick.
Okay.
if you're swimming, let's say you go to Cabo or you're down in Cancun, and you decide that you're going to get in that water. Shigella dysentery is very common in the waters off of Mexico. If you ingest 8 cells, so essentially in that case, getting water in your mouth or nose, the likelihood of you getting poisoned is pretty good. Because the threshold
wow.
for Shigella is only 8. So 7, you're okay. Don't ask me who figured that out. But 10, that's going to be a problem. With folks like with the brain-eating amoeba, you don't even have to get it in your mouth. It goes in through your nose, and it comes up to basically the frontal lobe, brain stem or frontal lobe sinus area. And once it gets there, it causes inflammation, which then causes the brain to swell, which, as we know, is a bad thing. So you don't even have to eat it. It just has to get into your nose, into the sinus, and that's when it starts causing problems. Out of the 305 cases, 1 survivor. And generally what that is poor water management, no chlorine in a pool, and or some of these bodies of lakes just have them in them, and folks are unlucky. But yeah, with a lot of these microbes, there is a threshold. And as long as you don't cross the threshold, it's okay to ingest, or at least that's what the FDA and our health departments tell us. All right. So believe it or not, just so that you guys have some info, and there's no reason to lie about it, the microscopes in the A&P and micro labs cost between $500 and $800. Right, they're your typical compound light microscope, and they would fall under the bright field area, where the organism is lit up around a very light field. The organism is typically darker than the surrounding brightness, and or the organism is usually stained so that there's contrast. There are some other really cool kinds of microscopy, and I'm not even talking electron yet, that use different kinds of light. And so in dark field, again, with the help of special dyes, usually fluorescent dyes, light up the organism brightly, but the surrounding field or the surrounding circle that you would see would be black, completely dark. And some really cool dyes here are used for those kinds of things. Phase contrast microscopy might alter the light slightly, differences in intensity. It might pass it through a polarizer a little bit. And so the light that's generated versus the light that hits the sample is a little bit different. And we use this to kind of highlight intracellular structures, inclusion granules, contractile vacuoles, those types of things. And I'm going to show you a picture of a paramecium. So you're thinking protista, you're thinking ciliated, and the top panel is going to be bright field, the middle panel is going to be dark field, the bottom panel will be phase contrast. And you'll see, even though it's the same organism, you're going to see 3 different views of it. And I'll zoom in a little bit for us here. So this is in fact the paramecium. You can see the contractile vacuole here under the light field. You can see the cilia kind of a little bit. The oral groove maybe right here. And then you've got some inclusion bodies and or full contractile vacuoles. Going under dark field looks real different, right? And the look of the contractile vacuole is very different. The cilia almost all but disappear. You can kind of see them. They look a little green. And the rest of the bodies, these kinds of structures pop up really, really easily. Versus phase contrast, where the cilia is amazing. The macro and the micronucleus here are very, very visible, but the contractile vacuole goes away. And you see a lot more of the granules and the inclusion bodies within the microorganism. And I did say that this particular organism does have a macronucleus and a micronucleus. And we totally would have missed that if we actually didn't have the phase contrast microscope. So that's kind of a really cool thing to look at. You really even don't see the nuclear structures here as much in the bright field as we do down in the phase contrast. So the bad news, these tend to be much more expensive. You need to purchase dyes and other types of chemicals so that you can see these more easily. And these microscopes can cost tens of thousands of dollars. So these are kind of labs that have grant monies and or do specialty work that have this kind of stuff. we start to go down a little more, and some of the cooler ones are the fluorescent microscopes or the lab tools that use lasers. A flow cytometer is one of those. And a flow cytometer is a device that usually shoots an organism with 2 lasers and uses the scatter of light to determine how it fluoresces and how much forward and side scatter. So what the size and the dimensions of the organism is. So lasers are really cool. In the case of fluorescent microscopy, it's a dark field again. And instead of using dye, we're using the lasers to excite the pigments in the microorganisms. So what you're not going to see here are regular eubacteria. An E. coli, a Staph aureus, you're not going to end up seeing. You're going to see bacteria that are photosynthetic, purple sulfur, green bacteria, green sulfur bacteria, bacteria that glow pink or magenta because of their bacteria rhodopsins. You'll also see phytoplankton, and they'll glow different colors based on their photosynthetic pigments. Pink if it's phycoerythrin, blue if it's phycocyanin, greens and such for other types of chlorophylls. And so what typically happens is, if I'm a phytoplankton biologist, if I'm out there in the open ocean, and I hit it with my lasers, I see stuff like this, right? And I see these things because they autofluoresce when they're hit with lasers. There was a little bit of a note here about this being useful in diagnosing infections. That is also very true. Some types of fungi in particular will tend to autofluoresce when you hit them with a laser, or something you have at home, or when you hit them with a black light. So if anybody's got a black light at home, and I don't mean to be dirty or grimy, plug it in and start walking around the walls. And I guarantee you, wherever there's drips, wherever you've splashed from shoes, wherever you've splashed drinks, you're going to find some kind of fluorescence on the wall with the black light. If you go in the bathroom and you hit the grout and the tiles, I don't want to tell you where, but you know, be thorough. You'll obviously start to see the different fluids fluorescing. If you don't want to go that route, just look in between the webs of your fingers and you will see autofluorescence there. Fungi are part of our natural and normal flora. It's the overgrowth of fungi that we don't like to see. And the last one that's kind of interesting, and I don't want to creep anybody out, but it's just what it is. Ringworm. Anybody ever have it? I'm not ashamed. I got it. I got it from my cat. Also not ashamed to say I have cats. I'm ashamed to tell you how many I have, so we're just going to skip that part. But one of my cats went, sorry?
I said, I hope it's not over 2 cats.
It is more than 2 cats, yeah.
Oh no.
Look, I'm not a liar. It's 7 3/4. And I just lost 2 kittens to a storm outside. They disappeared after the snow. Only the one strong little boy is out there. So I may have to take him in too and find him a home because I don't want to lose the 3rd one.
Oh my God.
But I have 7 3/4 because I'm a sucker and my wife is a sucker. And when they're hurt and they're injured, they come in and we're just terrible fosters. One of them has 3 legs, not 4. That's where the 3/4 come from, in case anybody's asking. There's still 8 mouths. So one of them ended up going to the vet, and she came home with something that she didn't go with. She was immunocompromised. She was a hot mess. Her fur started falling out, and we said, Christmas, what is this? And so before we knew what was going on, I have a cat, he's 18 and a half now, who's orange, who interacted with this cat, and he started to have all his hair fall out. So I got my black light, because of course I had one as a college student, and it's still in my house. And I turned it on and I put it over the orange cat, and he started glowing green. So what the vet did was grab a woodman's lamp. I grabbed the UV light, and ringworm, that particular fungus, glows green under the black light. So then, of course, I started putting it on me, and what I thought were mosquito bites were actually ringworm. So I said, okay, and I started myself on Lotrimin, right? And about a month it went away. But the lasers and different wavelengths of light can be actually very helpful to diagnose certain kinds of fungi or certain kinds of infections without the need for anything more invasive. If I had gone to my doctor, they would have taken a skin scraping. They would have wanted a culture. It would have been a couple of days to a week. I just put the black light on and went down to the CVS, right? And the itching and everything else and just how it looked was really the diagnosis for me / The cats definitely had it, so of course I have it. But it's kind of interesting. And if you Google Woodman's lamp, it's essentially a longer wavelength light that reacts very, very handily with a lot of fungi that you might find in the forest. And that's where this Woodman's lamp is really coming in handy. Now, of course, the last piece of this, and we'd be remiss without speaking about it, is the electron microscope. Oh, and by the way, the fluorescent microscopes run upwards of $30,000 to $50,000. And the flow cytometer can run in the hundreds of thousands of dollars, same as the electron microscope. The electron microscope can be, $300,000 to $500,000, which is why we don't have one at the college. Ramapo College has one. I've never been allowed to use it. Purchase has one. If you take the course, you can use it. The folks over there are pretty friendly. They let us exchange samples sometimes. There's one at Binghamton, if anybody cares. Maybe for a BSN, you guys get to use it. There's one at Cornell. And where's the other one that I know of? Queens College. There's one at Queens College. There was apparently one at Concordia, but they are no more. They have been absorbed by Iona. So we don't know where that is going to end up just yet. Nassau Community happens to have 3. And they have 3 because they had a very strong department chair who wrote a lot of grants and spent a lot of money. They also have 20,000 students, so they have quite a large biology department. And Rockland Psych also has an electron microscope. Theirs is a little older, so if you guys go down there for psych nursing, there's one that takes up actually 2 rooms. It's one room for the sample, and it's the other room for the machine and the prep. So these can be quite large, oftentimes taking up at least half a room because of the generation requirements. And as it sounds, it's literally electrons that go through the sample. So the samples are vaporized by taking a picture or an image. And what you typically have to do is take a sample that was alive, cut it very thinly, and coat it in some sort of metal. A lot of the time it's palladium or platinum. And then you put this very thin and prepared sample in the path of the electron beam, and you zap it. And in doing so, the computer detector receives a lot of information. A lot of paths of these electrons do not get obscured. They do not scatter. So you get amazing resolution with amazing detail, 100,000 times X magnification, maybe 1,000,000 times, depending on yours. And now with computers, there's also post-image processing. And so the stuff that you end up seeing, you can magnify something that's a nanometer and it looks like a cheek cell that you would have seen in the lab. Now there's 2 different varieties of these things. One of them is called the transmission electron microscope. So a TEM, transmission electron microscopy, if you prefer, or scanning electron microscopes for scanning electron microscopy. And so transmissions tend to be a still picture, while scanning tends to go back and forth and recreate a 3-dimensional version of what you've just irradiated. If you guys remember in A&P, I'm sure you saw the theory of the plasma membrane and the Davison and Donnelly model before you got to the Sanger and Nicholson model. And one of the things they actually took electron microscope pictures of was the phospholipid bilayer as it was peeled apart. And you could actually see the intrinsic proteins and the extrinsic proteins fitting together literally like negatives when they peeled the 2 layers apart. So the electron microscope was quintessential actually in finishing the phospholipid bilayer or the fluid mosaic model. Now what these tend to look like are really big things that are nondescript unless you took the picture or know what you're looking at. And so I don't plan on giving you these for tests, but I want you to see these because they're really cool. So what we've got here are copies of some electron images. And what it looks like to me here are some cells with either viruses on the outside or those are viruses with protein spikes. And I'm leaning toward these are viruses without the key. And so all of these things on the outside would actually be protein spikes. Or if we're looking, if we're looking inside the cells, that's the same paramecium under an electron microscope. And you can label individual cells, individual granules inside of this, realizing that this is another one still, this is another image of another organism yet. So really, really detailed. Ribosomes, you can get to the level of ribosomes. You can see polyribosomy happening. You could actually see, and you know what, I'll find it in a hot second. You could actually see viruses infecting cells. And in fact, I actually want you to see that. Because for me, it was always one of those questions, how many viruses infect a cell? I don't know, how many? Well, is it one? Is it more than one? It was really hard to tell. And it wasn't until I went to a seminar where they started doing this with fluorescence that you really had an appreciation of how many cells and how many infections it actually took. So I'm going to switch over to just a different image real quick so that you can see this. That's an E. coli being infected by a virus, right? And those are technically bacteriophage on the outside of the E. coli. And all of these dark pieces are viruses. This is a virus, 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 virus. And even all of these things down here are viruses. So if you were asked, it's probably hundreds of viruses infect each cell. And so when you go to bed feeling good, you wake up in the morning with a cold, when you've got an army like this of bazillions of viruses attacking the epithelial cells in your nose, or the coronavirus that's part of the common cold in our nose, or rhinoviruses, it's really easy to see why 6 hours later you're destroyed, whereas 6 hours earlier you felt great. Right? The TikTok video where the young lady gets that triple mocha monstrosity from whatever coffee place she was at and sits there and says, I can't taste this. This tastes like nothing. I should taste it. She said she could taste breakfast, but she couldn't taste the mid-afternoon coffee. With an attack like this, you understand how it happens so quickly. And I was blown away. I wasn't really sure how many it was either, to be honest. But I guess, you know, if it takes a 100 million E. coli to make us sick, it probably would take a bunch of virus to make us sick. I guess that makes a lot of sense. This, though, is the capper. That's a scanning electron image of a paramecium. Wow. The cilia, the oral groove, the openings, the detail here is fantastic. Other microbes, other cells, these are probably bacteria that are living in association with this organism. Tremendous, isn't it? And for the low price of $300,000 to $500,000, you could do this too. All right, that's also why that not every college has one. That's why this stuff is spread around. But you can see for folks who are doing this for publication, for teaching, they only need to take this picture once. And I always tell my general bio students, if you can operate that machine and you can generate a picture like this, you could sell this kind of a picture to Tolero, to Reese and Campbell. And maybe you like science, but you're really more of an artist. Here you go. Right? This is an actual image that's been colored by computer so that you could have the benefit of not having an electron microscope, but still get to see what they see. Amazing thing. All right, what do we have left? Sample preparation, those are fun, and staining. So two kind of quick things, nothing terrible. Oh, let me zoom out for you guys. Again, this is part of the lab that I'm sorry you guys are missing this semester. Sample prep is different in a lot of different situations. So in A&P, most of the things that we gave you were already made. Back 50 years ago, my father used to make tissue mounts. And so they would use a microtome and take really thin slices. They would stain them, they would put a cover glass on, and with their hands, dip it into a solvent to seal it. And that solvent most often was xylene, according to my father. Now, also, as you guys know, xylene is a mutagen. And so now that my dad is in his upper 70s and he's been exposed to these chemicals for 30 years at least, he has very strange pigmentation patterns underneath his nails, right? Melanin is starting to grow. And those fingernails grow faster than the fingernails on the hand he did not dip in xylene. So there was obviously some weirdness that happened. We do still make wet mounts today. We tend to use depression slides that have a reservoir for some of the sample. We tend to use cover glasses. We would tend to use clear nail polish to seal slides rather than using things like xylene that cause chemical melting or reactions of cover glasses. In the lab that we're not doing, unfortunately, you would make fixed mounts. So you would take a loop, you would sterilize it, wait for it to cool down. Right? And by the way, waiting for a loop to cool down, and I wish I could see what I'm holding up here, hold on. Right, doesn't mean waiting for it to cool down because that defeats the purpose of heating it up. don't none of this either while you're waiting. You just want to hold it and let the red color go away. Count maybe 20 Mississippi before putting it into the tube. If you put it into the tube and you didn't wait long enough, you hear, right? That's basically mass murder. You have to heat the loop again, let it cool, and then go back into the sample. Take out a small amount. And when I say small, less is better. So if you went in there and you pulled out the loop and it didn't have any liquid in it, even better still. You touch it to the slide and you start to make a smear. And that smear is nothing more than you hitting the glass slide and making about a nickel-sized smear or a spread. You're then going to let it dry. And the reason why you let it dry is because if you go right to the heat fixing step, you actually boil the cells. They actually explode. So you let them dry, and then literally you take your slide. I guess that shows up, right? I think that shows up. Yep. And if this is the Bunsen burner, you would literally 1, 2, 3. You would just wave it over the top. And you don't have to get into the flame. You just have to be near the flame. And the heat from the flame will fix or attach the proteins from the cells to the glass. From that point on, they will not come off. And you would then go on to your staining and or your microscopy piece here. In terms of staining, there's a bunch of different kinds of dyes that we use. The cationic dyes are basic, but they have positive charges. I probably should show this to you. The anionic dyes are acidic with negative charges. And so you typically would use the right stain for the right situation. And that's going to change a lot depending on what you're looking at. Flagellar stains are very different than cell wall stains, are very different than spore stains. Obviously, you would have an idea of what we were getting ourselves into before you chose a particular kind of stain. And simple stains, literally just one kind of dye, really could be anything. I mean, quite frankly, strawberry juice makes a good stain. Beet juice, if you filtered it, makes a really good stain. In the lab, though, simple stain is good enough just to see the cells. All you're going to learn about it is the morphology. Is it a coccus? Is it a rod? Is it a spirilla? That's it. The differential stainings, so gram staining in particular, and acid-fast staining, allow you to use a bunch of stains or chemicals in a very specific order to learn something about the chemistry of the cell wall and then something about the chemistry of the organism. And so differential stainings for the gram stain method involve crystal violet as a primary, involves iodine as a mordant, so a killing agent, some ethyl alcohol, and a counterstain, which could be a lot of things, but we tend to use safranin. In the acid-fast version, our primary stain is carbylfuchsin. We have to heat it. We use alcohol with some hydrochloric acid in it, and we counterstain with either brilliant green or methylene blue. So differential staining is a stepwise process that allows you to learn about the chemistry. And if you're in a doctor's office or a hospital and they do a quick gram stain, they will know exactly what kind of antibiotics not to use in terms of starting your treatment. Broad spectrum, gram positive, no problem, we know what to use. Broad spectrum, gram negative, we got you. And so this is really useful without having to do extra steps. And if we're wondering what some of this stuff looks like, zoom out a little here. There's a simple stain here, just crystal violet, so it's a nice purple color. And you see E. coli, which tends to be some stubby rods. In the middle, I guess it's pane C, you see gram staining. And we've got, let's see, some diplobacillus, it looks like, the purple. And it looks like we've got some bent rods or maybe even spirilla when it comes to the gram negatives. A little bit of a hint here, we don't have any Vibrio, we don't have any spirilla or spirochetes in our lab. So if it looks spiral, it's probably not. It's probably just a rod of some variety, right? We don't have those to show you. Let's see, there's an India ink stain here. So these are of Cryptococcus, and it's more of a negative stain. The Cryptococcus is the white, the India ink is the dark here. Let's see, this is a Corynebacterium, which is different than coronavirus. Corynebacterium typically are throat or skin infections. And what you've got here are these little stubbies that are in diplo form. So they're tubed together. Let's see, on the right, you've got some flagellar stains. The one in the middle here, the acid-fast stain, is the primary diagnosis for tuberculosis. And so what you're seeing is those pink rods. right? Which are known as acid-fast rods. They're not positive, they're acid-fast. Even if you're a clinician or you're a nurse and you're running a sputum sample and you see just one pink rod, you have to tell the patient the bad news and start them on TB drugs. All the stuff in the background, all the blue stuff, we don't care what it is. It could be a rod, it could be a coccus. They're non-acid fast because they did not retain the pink stain. You do not care about any of that stuff. None of that matters in your investigation. You want the pink rods. Well, you don't want the pink rods, but you want to find them if they're there. The reason why it's acid fast for the tuberculosis over the gram stain is because the acid fast organism has a mycolic acid wall. So essentially, you have to penetrate a thick, waxy layer, and the crystal violet is just not up to the task. Carbofuchsin is stronger. It has phenol in it. And further, you heat it so that the phenol can penetrate the mycolic acid. There are other methods to do this now that don't involve heating, but you definitely need a hefty stain. Gram staining doesn't do this justice. And then lastly, you've got some spore staining here. And you've got a negative stain on the left. And as always, like our microscopes, we're talking about the right tool for the right job. There's definitely a time and a place to use these. And as you get through the exposure in the lab, you start to see and you start to figure out and learn when is best on a simple stain versus when you really want to go gram stain or acid fast. And again, the setting and the contacts will help direct you. And so, with that, how we doing? We're still alive. Everybody's still here. That's a good thing. That was probably a long, almost 2 hours, and I'm certainly not going to prolong this. But we should talk some housekeeping while we're here. So I'm definitely going to put this up. I'm going to render it tonight. I'll have it uploaded tonight. YouTube is a little backed up. I think it will render in standard definition. It might take a while for the high def to come, but that's okay. Next week, we would be starting prokaryotes. And I kind of would still like to do that on Thursday. Next week, though, we are supposed to have test number 1. And so what maybe I would like to suggest, and we can fine tune the plan here, is to open it up on Wednesday, do maybe an hour and a half of lecturing on Thursday / office hours on Thursday after that, and then leave the test open through Saturday. So folks who wanted some extra time to study could. Folks who wanted to take it right away and only have class time, you still get an hour and a half or 2 hours to do it. And this will help to accommodate your work schedules. And it will also give me like a half a week of buffer so that should things happen, should we need to go through more details, folks ask more questions, we have a little bit of buffer so that we can continue and do everything that we need to do without being worried about time. So yeah, Mario, Wednesday, probably 8 a.m. through the close of the day on Saturday. Right, and there's definitely enough questions that I have where it's unlikely that anybody's getting the same test. So for that reason, I'm not really terribly interested in honor lock. I know folks have asked. These aren't meant to be collaborative, so obviously keep that in mind. If I see strange wrong answers and strange wrong choices on a bunch of tests, I ask those questions. Yep, the test will be the first 2 classes and the chapters that correlate to that in whichever book you have. So Tolero, it's like 1 and 3 on the older edition. Your lab manual, it's a couple extra. How many questions? I want to say between 40 and 60, 40 and 50, somewhere in that range. So it should take, I don't know, an hour, hour and a half-ish.
Is there any areas we should focus on?
I don't want to say there's anything particular. I would say, you know, the scientists are good to know. A couple examples, probably not your primary focus. You know, some stuff about biotech, some stuff about the kinds of microbiology would be helpful. Some stuff about stains, some stuff about microscopes, some things about culture. You know, the 5 eyes would be stuff to take a look at. The test does need to be completed in one sitting. So whenever you do plan to log in, make sure you get it completed. Make sure you have good internet. Make sure that folks aren't trying to play Xbox while you're in there. Do the best you can with it. Try not to use an iPad. Try not to use your phone, certainly. They have unstable internet. They do funny things with Amazon Web Services. And what I tend to see, if folks have multiple instances of Blackboard open, so this is just kind of if you guys tell me you got kicked out, I know what happened. What I see happening there is you'll click into the exam and it will automatically submit it for you because of the other older expired Blackboard window that's open. And so you'll fill everything in, but you'll get a 0 out of 0 or a 10 out of 100 because it auto submitted due to the second browser. So try to use Chrome, try to have one window open. It just makes things smoother. If there is an issue, and we'll obviously talk about this next week on Thursday, if there is an issue, email me and I can look at it and see what happened. If it was a browser issue, I can open it back up. If it's something where you just didn't like the grade and they're all filled in, I'm not opening it back up. And there is only one attempt. So this is not like a mastery type thing where you keep doing it till you get 100. There's just one attempt. And yes, it is going to be timed. I'm thinking it comes into 75 minutes or so between 75 and 90. I don't know exactly how long I'm going to set it for because I need to see how many questions I pull. But if I keep it to 40, I'm probably looking at 75 minutes, give or take. If it's more than 40 or 50, then I'll probably bump it up to 2 hours. But I'll see what I end up grabbing and what looks good this time. In terms of where you'll find it, nobody ever asked me that, which is kind of weird. Under Blackboard, under content, there's a folder that says tests. And so it's completely blank. You don't see anything in there. Wednesday at about 8 o'clock, you'll actually see test 1 appear. All right, and so it'll appear 8 in the morning. At least that's what I'll set it for. It will stay open until Saturday at 11:59. That's a great question. So the grades will be posted after everybody is finished. So you're not going to get instant feedback. Unfortunately, that's not like a scratchy lotto. And what you'll have to do after the test is go in and click on grades, click on the actual score. It'll show up in blue, showing you that it's a link. And that will take you to what you picked, the question, and what the answer was. So hopefully we see all greens, right? Green pick, green answer. So by Sunday morning, you'll see the grades. Oh, yeah, good question. Multiple choice, true, false only. I did not put multiples in there. I didn't do the select all that applies, although I have it on good authority that nursing likes to use those questions, and I probably should be.
Please, God, don't.
Yeah, I don't plan on doing that to you. But I know that is a favorite of the nursing department, so just be aware of that.
Oh, it is.
Yes, it is. Don't you hate that too? All right, I'm going to hit the
Sneaky.
stop button